All right. So good afternoon, students. Thank you for attending this lecture. This is lecture 16 in the syllabus, and we're going to go over really the topic here is homeostasis in fishes and really integrative, what I like to call integrative systems. So things like the central nervous system and how fishes operate. And there are going to be some differences here between different types of fishes that you'll have to take note of. And so, as usual, I'll answer quite any questions at the end of the lecture, and I'll post all these recordings online for you to access them. So with that, I'd like to just do some very brief introductions here of definitions and terms from like intro to biology. Homeostasis. The definition is, this is a process to maintain some kind of equilibrium. So remember that your organism, whether it be a fish or a human, is always battling against its environment to maintain some kind of equilibrium. And this is a never-ending battle. And this allows for physiological processes to function properly. So you have to maintain the inside of the organism in a state that is favorable to its existence, and this state is not the same as the environment around it. This includes things like modulating body temperature or regulating the temperature of the organism. Too hot is not good, too cold is not good. I always think of the Goldilocks, just right. Also, mechanisms of water, solute, and pH balance. You have to maintain the right salt concentration and the right pH for all your enzymes and proteins to work. You have to maintain your immune system so that competing organisms such as disease-causing bacteria and viruses do not infect you and kill you. And then you have to be able to maintain some balance following stress. So for instance, predator avoidance, so you're not eaten. There are many others that we don't have time to discuss, but in this lecture, we're going to focus on these four major components, and these also are detailed in the reading of your book. So we're going to begin with two major systems that control or essentially regulate all these processes, and that is the endocrine system and the nervous system. And so a lot of this is going to be review. The endocrine system is defined as a network of glands that release chemicals called hormones into the blood. An endocrine gland is a ductless gland with a blood supply. The hormones are transported in the blood. That's how they get to their destination of action. And the products of these glands are called hormones. They're secreted into the blood where they then act on specific tissues. So hormones are chemical substances produced in these ductless glands, carried by the blood to other parts of the body where they exert their effect. So that could be the muscle, for instance. That could be the gonads for reproduction, but it has some kind of target destination that it affects. And it could be also the entire organism. Like for instance, if you want to regulate your metabolic rate, which also might be involved in body temperature regulation, a hormone is most likely going to control that process because the metabolic rate involves the entire organism. Key things to remember: hormones do not supply energy to chemical reactions. They do not do this. They are very potent. Only a very small amount of hormone is needed. A human male, all the testosterone in a human male's body can fit on the very tip of a pin. It's a very potent hormone. And so not a lot of it is needed. They are very rapidly removed from the blood. So once these things are produced by these ductless glands and dumped into the blood to exert an effect, they are rapidly removed so that the signal does not persist beyond its need. They do not supply energy to reactions, but they do regulate the rates of chemical reactions. So for instance, the speed. They can speed up chemical reactions or slow them down. And overall, they generally act to maintain the internal environment, i.e. this homeostasis. They integrate with physiological events to coordinate things. For instance, if you're increasing your growth rate, you might want to increase your food consumption rate, right, to provide enough energy to pay for that increased growth. And they can control growth and development of body parts. For instance, growth of body tissues like muscle, but also, sexual maturation, production of gametes, reproduction, spawning behavior, things like this. And so they coordinate a lot of these activities. And because they act globally on many different tissues, they're a perfect delivery system for this control. Now, the nervous system, on the other hand, consists of some kind of central processing center. Now, I'd call this a brain, but higher fishes have a brain, but a hagfish is really going to have something more like a cerebral ganglion. Not as sophisticated. Again, these things are tumbling around in the muck on the bottom of the ocean with a single semicircular canal to let them know their orientation, but they're not as complex at processing things. And so collectively, we would call any of this some part of the central nervous system. So dry fishes will have a brain. Agnathans oftentimes have a ganglion. It functions in the same. These, along with nerves, bring information. What we call afferent neurons bring information to the brain or the ganglion in the central nervous system. And then efferent neurons send information or actions out. So for instance, when the eye sees an image, an afferent neuron through the optic nerve will send that message to the brain. The brain then will send a message out. For instance, if the eye sees a predator approaching, the brain then sends an efferent signal out telling the fish, you might want to swim away from that. And that then tells the muscles to move. So the functional unit of the nervous system is a neuron, and information that is transmitted in the nervous system through the electrical activity of these neurons, which are interconnected. Again, sending signals away or towards the central nervous system. So signals are transmitted between cells by the release of chemical neurotransmitters. This sort of tells when one neuron releases this chemical, it tells the neighboring neuron, hey, you should release that chemical and continue to do that downstream. And this then enters that synaptic space between the two neurons or an effector cell, for instance, again, a muscle. When a neuron innervates a muscle, it releases a neurotransmitter that tells the muscle to contract, for instance. So pretty straightforward. Now, when we compare and contrast endocrine and nervous control, the first thing is this: the nervous system operates very quickly. The target may be pinpointed. So for instance, I can move my muscles like this very quickly. I can also, my brain can tell just my bicep to move, right? So it's pinpointed and it's short in duration. This is very quick. High-speed signal transmission of the nervous system makes it ideal for controlling skeletal muscles, for instance, especially when speed's important, rapidly escaping a predator, for example. Compared to the nervous system, the endocrine control is much more slow. The target is a general cell population, so it's not pinpointed usually. It could be the whole organism, like controlling bioenergetics. And the influence is longer in duration. So for instance, when we talk about gametogenesis and reproduction, the endocrine system might tell the female fish to produce eggs, and it takes the fish months to produce eggs. So this hormone signal can persist in the organism for months while the female produces eggs. Whereas here, the muscle contraction occurs in a fraction of a second. So very rapid, very long, slow term, or very slow long term. So the slow global effects of hormones from the endocrine system make them ideal for coordinating widespread physiological processes. Examples are growth, sexual maturation, and metabolic rate. Okay, so those are two major concepts there. When we look at the nervous system and moving forward here into coordinating physiological activities, it's divided into the sympathetic and parasympathetic systems. Now, I like to think of these as having opposing activities during a fight or flight response. So what this response refers to is an alarm response where a predator, for instance, suddenly appears and the fish must decide immediately whether it needs to fight or run away. That's kind of funny. I'd say probably swim away, not run away, but this is how it's typed. Sorry. In this situation, the sympathetic system swings into action and, for instance, would direct blood flow to the brain and skeletal muscles, because if you're trying to avoid a predator, you need to swim, and that's your muscles that are doing that, and your brain needs to find an escape route. You really don't need to be digesting food at that time. So, for instance, we might borrow blood from the GI system and send it here. This is one nervous action. The adaptive advantage of this response is that the brain and muscles have plenty of oxygen and nutrients to fight or to swim away. The GI tract, it doesn't need to be actively digesting at that point. You can go ahead and swim away, and once you're safe, then you can continue to digest what you just ate. Don't worry, it's not going anywhere. It's inside of your gut. So you can wait a few minutes. So the opposite of the fight or flight response then is the response after eating a large meal, for instance. So after ingesting something like you've just eaten and your gut is filled, the parasympathetic system then is activated. And this might say, you know what, there's no threats out here. Let's go ahead and direct blood to the GI system to aid in the digestion and absorption of activities. And then this operates when we call times are good. So there's no threats. It's time for us to relax. Let's go ahead and digest that. And so that's when it operates. And so when we say they're equal and opposite, you can sort of see this. Whenever the sympathetic system is activated, the parasympathetic system is deactivated, and vice versa. They are equal and opposite. So blood flow to the brain and skeletal muscles will be reduced after ingesting a large meal. And people talk about, like, after Thanksgiving dinner, you feel tired and sleepy. It's part of this. So that's what's going on here, parasympathetic system and sympathetic system. So with that setting the stage, now we're going to talk about some actual processes. So here are some terms to know. We're going to talk about thermoregulation, one of our first homeostasis topics. An ectotherm is an organism that relies on external sources to control its body temperature. Okay. Now, some people say that this is cold-blooded. It is, and some people say it's cold-blooded is not as advanced as a warm-blooded organism. Well, let's discuss what the differences are. The first is there's a couple strengths here. If you're an ectotherm, you've got a lower metabolic rate than an endotherm, and that means you got more energy that you can use to grow, for instance. You can also have more energy to reproduce because you're not having to maintain your body temperature. You got less energy required for maintenance because making body temperature actually, again, is not free, just like anything else. You got to pay for that. And so ectotherms can survive on less food. They don't need to consume as much to get more out. Okay. Examples of ectothermic vertebrates: reptiles, amphibians, and some fishes. Some fishes are outright ectotherms. Now, here's an endotherm. Now, endotherms generate and retain their own heat. So this is a high energetic cost because, again, generating heat is not free. You got to burn energy to do it. And this requires you to consume more food than an ectotherm because this is not free. This leaves less energy overall for growth and reproduction and other housekeeping functions, for instance, homeostasis functions. But a benefit here is that an elevated body temperature keeps thermosensitive reactions always operating optimally. When you're an ectotherm, your enzymes might not do so well at a really cold temperature, but they also might not do so well at a very high temperature. Whereas endotherms sort of can control this temperature, which means their enzymes they're paying a price to make sure they're always operating optimally. So examples here are mammals, birds, and a few fishes. We've already talked about these, things like tunas. Most fishes, however, are what we call poikilotherms. Now, this is an organism where the internal body temperature varies considerably. And so it's a type of ectotherm. It also could be a type of endotherm as well. The key here is that the body temperature varies considerably, whereas an ectotherm, like an ice fish that lives in the bottom of the ocean in the polar caps, is going to be living its life almost continuously at near freezing water temperatures. There's not a whole lot of water fluctuation or water temperature fluctuation there. Likewise, even though its body temperature is determined entirely by its environment, likewise, a tropical fish living at the equator is also an ectotherm, but it might not see a whole lot of fluctuation in body temperature because it's 80 degrees all year long. So they are ectotherms, but their body temperature might not vary throughout the day or vary throughout the year. Most fishes are, however, temperate. And so they are what we call poikilotherms, where their body temperature can vary considerably, and this can be throughout the day and also seasonally. A largemouth bass in Lake Jordan during the summertime has got a much warmer body temperature than a largemouth bass in Lake Jordan in January. Okay, so their body temperature varies considerably throughout the year. This is a consequence then of the natural ambient temperature. So they may remain temperature constant environments, for instance, tropical fishes or the Arctic fishes, but most of these vary considerably. So examples here are reptiles, amphibians, and fishes, and I would say most fishes are poikilothermic ectotherms. If I were to classify them, and you might have to know this for a test, I'm not sure, but if I were to say what a typical fish is going to be, or you could say an ectothermic poikilotherm. That's what a typical fish is going to be. So that's what most fishes are. Now, there are some exceptions. So the tunas, the mackerel sharks, family Lamnidae, the thresher sharks, family Alopiidae, they conserve heat generated by active swimming. And mussels then maintain body temperatures above their environment or ambient water temperature. So the bluefin tuna, for instance, mussel temperatures are 28 to 33 degrees centigrade, while the water temperature is somewhere between 7 and 30 degrees, or 7 and 30 degrees centigrade. So they are pretty warm-blooded. Not as warm as we are. We're about 37 degrees centigrade, but pretty warm. Yellowfin tunas maintain temperatures about 3 degrees centigrade above water temperatures. And so these are more of a poikilotherm. So depending on what the water temperature is, they maintain a little bit higher than that water temperature, but they don't really vary, or they're only about 3 degrees centigrade above that. Whereas tunas are less of a poikilotherm because they operate in a pretty narrow range, 28 to 33 centigrade. These fish do this in a similar principle to countercurrent water exchange with the blood that we saw in the gills last time. But instead, the plumbing of the blood vessels, the arteries and veins, are in opposite directions, such that heat is being transferred between the two pipes, the veins and the arteries. So blood flows towards large swimming muscles near the body core and passes through a network of small blood vessels in a Rita mirabile type structure. And this then runs countercurrent to blood leaving the muscles, which has been warmed by that muscular activity. And then heat in the blood leaving the muscle then is transferred to the blood flowing into the muscles, and thus it is retained locally and not lost to the environment through the gills, for instance. The gills are going to have a lot of heat leaving them. So what this does is it retains warm temperature inside of the muscle based upon the blood flow there. So blood flow typically goes heart, gills, Rita mirabile in the muscle, to the swimming muscles, to the heart, then to the gills, and the heat is lost. But this is what's needed to do in order for us to have oxygen exchange. However, if we follow the path of the heat goes from the swimming muscles to the Rita, back to the swimming muscles, to the Rita, back to the swimming muscles, to the Rita, et cetera, et cetera. And so the heat is circulated within the muscle. So when you look at then the plumbing, this is sort of what it looks like. A typical fish has these blood vessels that run down the trunk of the body here coming from the heart to the lungs, then down the trunk of the fish, where then it goes out towards the surface of the animal, where it's going to lose heat. And then that cold blood, because the heat gets lost to the water, the cold blood then circulates back, and this cold blood runs right alongside the warm blood, so it cools the fish down. There's no way for this heat to be retained. Whereas we looked at a warm-blooded tuna here, Thunus thunus, most of the blood from the gill is shunted towards cutaneous vessels near the surface and is carried through the heat-exchanging urethra. So heart to the lungs, and then these vessels come out like this, and here is this countercurrent urethra. So the warm blood coming back to heart exchange heat into these vessels, sending it right back out to the muscles. And see how that works. It's a circuit. So the warmth of the blood is circulated and retained here in the musculature. And so the reading has more information about this. When you look at other types of endothermy, there are fishes that have what's called regional endothermy. And this is maintaining elevated body temperatures only in certain parts of the body. And so oftentimes it's the brain. Or we talked last week or last time about how the eyes are a very energetically demanding sense. And so we can heat the eyes up too, which helps the eyes operate optimally. And so this permits utilization of colder habitats without suffering negative effects of temperature fluctuations on the central nervous system. The colder you are, the slower your biochemical reactions and the slower you operate. And so if you warm your brain up, you can think faster, right? So these fish are a little bit more astute, if you will. Also, their eyes work very well. So that means that their sense of sight is a lot better than other typical fishes by warming the eyes. Now, what happens here is there are ion exchangers in muscle cells. They're modified muscle cells. They don't necessarily, for instance, cause muscle contractions, but they sort of quiver or vibrate. Kind of like if you're really cold, you shiver. That's sort of what they do. And when they shiver like that, they produce heat. And that heat then is what we call a heater tissue. That heat then heats localized tissues, be it the eyeballs themselves or some parts of the brain. And in a very similar mechanism, it's the same reason why when you're really cold, you shiver. It's your body's way of trying to increase your core body temperature. Because every time a muscle contracts, it generates heat. So fishes will do this, especially some of these bill fishes, for instance, the brain heaters. So the swordfishes and the sailfishes. Other considerations of temperature are this. There are different responses depending on whether the temperature increases or decreases. So recall from the respiratory gas exchange lecture, dissolved oxygen levels in water are decreased due to gas solubility, partial pressure in high temperatures. So the warmer your water is, the less oxygen it will contain. So this is an outright major consideration, right? You have to be able to deal with that. It's actually paired with the fact, it's even worse, all of your chemical reactions increase the rate. So that means all of your chemical reactions are speeding up, which means you need more energy. In order to burn more energy, you need more oxygen, but you've got less oxygen in the water. It's sort of a vicious cycle here. Also, at really high temperatures, 35, 37 degrees centigrade or higher, some of your proteins in the fish might start to denature. And so there are heat shock proteins, which are a group of proteins that are synthesized at high temperatures. And what these do is they help protect proteins from denaturation. It prevents them from unfolding and becoming damaged. And so these heat shock proteins are produced, and they help protect your enzymes so that they continue to function and they don't die. Now, at low temperatures, there are some considerations as well. The rate of all biochemical reactions slows down. And what this means is a predator can sneak up on you and eat you, and you literally don't have the energy to escape. And so you're at risk. At very low temperatures, also, intracellular ice crystals can puncture cell membranes. You can actually have ice crystals form inside of the cells. And of course, this leads, organelle death leads to cell death, which leads to death of the fish. So it's not good. And we'll talk about this importance of this in the next slide. In order for some fishes to deal with this, saturated and unsaturated fatty acids that comprise the cell membranes become altered at lower temperatures. So anyone's ever heard that marine fish have higher levels of omega-3 fatty acids? It's true. And the reason being is that oftentimes marine fishes, especially like the Pacific Ocean, is really cold, right? Cold water fishes have high saturated or have high unsaturated fats, like omega fats, because saturated fats produce very stiff, viscous cell membranes. And at cold temperatures, these become brittle and can break. And the cell can actually rupture and leak its contents out. So in really cold water, fishes actually use a lot of omega fatty acids in their cell membranes because unsaturated fats are liquidy and less viscous at cold temperature. And so this prevents those cells from rupturing. So cold water marine fishes will do this as an adaptation to protect themselves in cold water. Now, as they swim into warmer water, they can actually turn over the composition of their cell membranes. And so this is a transient kind of thing. And so in cold conditions, the cell membranes are mostly unsaturated fatty acids. In warm environments, they're mostly saturated fatty acids. And this is an adaptation. Other adaptations in the extreme cold. This is actually kind of an interesting thing. So fresh water freezes at about 0 degrees centigrade. But if you recall from chemistry, fresh water is densest at 4 degrees centigrade. And that's because ice is not the densest form of water. It's actually the densest at 4 degrees. Ice, because it forms these crystals, the molecules actually set slightly farther apart than they do in the crystalline structure than they do at 4 degrees centigrade. Ions and other solutes in the intracellular fluids of freshwater fish drop the freezing point of the fish itself to about negative 0.7 degrees centigrade. And so this means that a freshwater fish is never in a threat of freezing solid, if you will. The water around the fish is going to freeze solid before it does. And every now and then this will happen. Shallow ponds, there's an ice over where the fish will literally become encased in ice. But typically speaking, it's not like they have to worry about freezing solid because the water around them is going to freeze solid first. And this image sort of shows a sheet of ice forming over a pond, and these bluegill can survive underneath the ice. They're very slow. Their metabolism slows down. They're not very active down here. They're almost in a state of suspended animation because this water is usually about 4 degrees centigrade. So they just sort of sit there. So they don't have to worry about freezing solid. Saltwater fish, believe it or not, do. They can literally be swimming along and all of a sudden swim into ultra cold water and freeze solid. And that's because salt water freezes at about negative 1.8 degrees centigrade. This is below the freeze point depression of body fluids, and whether it's a freshwater fish or a saltwater fish, the body fluids freeze point depression is about negative 0.7 degrees centigrade, which means the fish, this is still, this is where the freeze point is. A fish can swim into water that's negative 1 degree centigrade, which means the fish itself will freeze solid, even though the water is liquid around it. And that's because salt water contains solutes, which means that it can become super cooled. It's still liquid because of the salt in it, but it's super chilled, meaning that if a fish swims into it, the fish freezes solid. So some fishes deal with this by increasing the concentrations of ions or other solutes in their body fluids, such that they actually reduce their freeze point depression to below this here. This means then that the salt water is going to freeze into a block of ice before they do while swimming in it. Also, some fishes use what are called antifreeze proteins. And what these are is they They are produced by the liver of some fishes to prevent the formation of ice crystals. So these are produced by fishes in the deep, cold water, oftentimes the Arctic waters or the Antarctic waters, and it then further reduces this freeze point depression so that the water around the fish is going to freeze solid before they do. So they have no threat then of dying this way. This brings the freeze point of the Antarctic fishes, for instance, of the body fluids down to that below this. So their freeze point depression then might be -2 degrees centigrade, and so they're safe. They're safe from freezing. So notothenioid fishes utilize antifreezes and increase osmolytes. And so with that, I'm going to show 2 videos. The first here is going to be one on the warm-blooded OPA, and the second will then be on the cold-blooded Arctic ice fish to sort of show these kind of adaptations. So here is the OPA first. And this is a sort of an article done by CNN. So the opas are a warm-blooded style of fish, and again, you sort of see the video clip. They maintain their body temperature by flapping their fins, and again, muscular activity, and the muscular activity then generates heat. And so here then is a video on the ice fishes.
Every creature that lives in and around Antarctica has to deal with one overwhelming reality: the cold. Some use insulating fat and water-shedding feathers. But for ice fish living in below freezing seawater, the answer is antifreeze. Ice is everywhere, including in the bodies of the fish. But the fish have proteins that latch onto the ice crystals and keep them from growing and destroying living tissue. And scientists have recently found that these antifreeze proteins also keep the ice crystals from melting. To figure this out, the researchers dove beneath the ice and collected fish. They drew antifreeze proteins from their blood to test in the lab. They found that even at temperatures that would melt normal ice crystals, the ones in the fish don't melt. So ice crystals must be accumulating in the fish throughout their lives, although scientists don't know exactly what the negative effects might be. Internal antifreeze is obviously a lot better than freezing to death, but it is one of those evolutionary solutions that probably comes with a hidden cost.
And so that there are the ice fishes. And you sort of see that in the environment in which those ice fishes are living, if it wasn't for those antifreeze proteins, the water around them is colder than what their freeze point depression would be. They would actually freeze solid while swimming around there. The only reason why they're not solid ice is because of these antifreeze proteins. So very unique, very cool adaptation there for those kinds of fishes. So that's thermoregulation. Now we're going to talk about osmoregulation, which is another one of our topics of homeostasis. Now, there's a couple of key concepts here. The first is this. Osmoregulation, the first thing we're going to consider here is that pure water in unbalanced salt solutions are toxic to biological systems. And so when we look at this, osmotic or ionic properties, when they exceed limits, this is why these are toxic. When they exceed the limit and become toxic, they can change cell structure and morphology or shape. They can disrupt ion transport and cell permeability, which interferes with things like oxygen gas exchange or carbon dioxide transport in the blood. They can damage cell proteins inside of the cell, and they can also then cause cell death. And again, when we talk about cell death, it's usually organism death. So when we talk about osmoregulation, this is osmosis, right? If you have water in a solution, it is always going to diffuse down its concentration gradient. So for instance, water in a low salt solution, what we call hypoosmotic, is going to diffuse down its concentration gradient into a hyperosmotic solution, this high solute. Another way of saying this is low solute means high water, and high solute means low water. And so it goes down its concentration gradient. So knowing this, what organisms, including fishes, have to do is they have to do two things. The first is they have to regulate this water, and that is called osmoregulation. But in order to regulate water, they have to actually regulate their ions, and this is called ion regulation. And the reason why this is important is nowhere in life, there's no biological system out there that can pump water. You know, plants can transpire it, but proteins cannot pump water. All proteins can do is pump salts, ions. And when you pump ions in a particular direction, then water will follow it based upon osmosis. So, osmoregulation is defined as the struggle to maintain cells in a medium in which they can be bathed without a net flux of water across some kind of membrane. So this would be an isoosmotic situation where water doesn't diffuse anywhere because the salt concentration on both sides here are the same. That's osmoregulation. Maintenance of appropriate osmotic pressure both on the inside and the outside. Ion regulation then is defined as the struggle to control movement of specific ions across a membrane down their concentration gradient, a maintenance of appropriate ionic concentrations between the interior and the exterior part of the cell. So if this is the membrane, inside, outside. So this is primarily concerned with pumping some specific ions. Generally speaking, sodium, potassium, chloride, magnesium, calcium, sulfate, carbonate, and bicarbonate. And again, I talked last time about how most carbon dioxide in the blood is dissolved as carbonic acid, which then dissociates into a proton and bicarbonate. This is a very important one related to carbon dioxide production from metabolism. So we look at fish having to deal with this. There's a couple strategies. There's four major problems with maintaining water and salt balance. So osmoregulation and ion balance, ion regulation. The first is ionic regulation in an isoosmotic media. And this is sort of what hagfishes do. The agnathans do this. And what they do is they have to maintain salt concentrations in their blood, but their blood plasma, their osmoregulation, is identical to that of seawater. So the blood plasma of a hagfish is identical to that of seawater, same osmolality. Then you have what's called freshwater fishes maintaining a hyperosmotic state. This means that the blood plasma of a freshwater fish has a higher salt content than the water around it. Then there is maintenance of a hypoosmotic state. This is saltwater fish. This means that the saltwater fish has solutes in the blood that are lower in concentration to its environment, meaning that water is always leaking out of the fish and into the water around it, the marine environment. And then you've got the crazy lungfish. They live on land, so they have to deal with like outright dehydration. And so these are unique in the sense that they have similar structures to us, a very tough integument that prevents them from drying out, just like we have a skin that prevents us from drying out. Most fishes are concerned with 1 through 3 above, and for the following reasons. So freshwater fishes are always drowning. A freshwater fish has higher salt concentration in its body, means water always is entering by osmosis. They're drowning. Saltwater fish have a lower salt concentration in their body compared to the exterior environment, which means they are always dehydrating. This is a desert. The ocean is a desert, drying these fish out. These fish are being flooded. So there are a couple solutions to this problem. The first is, let's decrease the permeability of our surfaces to water. Let's reduce the rate at which water can cross these membranes. Let's waterproof ourselves. That's what the lungfish does. That's also what the fishes do. Their scales are the structures on their surfaces are designed to sort of prevent water from flowing across it. Really, most of the water exchange occurs across the gills, which we talked about is required because that's where you have to pick up oxygen and offload carbon dioxide. Second strategy, let's create a water pump. And this is really an evolutionary mechanism. This was created by freshwater fishes. It's called the kidney. And the original kidney was literally that, a water pump. Freshwater fishes are pulling in a lot of water every day. Let's go ahead and pump that water out. Let's produce large amounts of dilute urine, and the kidney does this. It's a water pump, pumps water out. And then also let's create salt pumps. Both freshwater and saltwater fishes do this. The salt pumps are located in the gills. And the only difference here is that because they're facing 2 different osmoregulatory challenges that are equal and opposite in direction, the pumps basically operate in a different direction. Saltwater fish pumps salt in, or freshwater fish pumps salt in, saltwater fish pumps salt out. So that's what they do. So when we look at these solutions, there's 4 different modes of what we call hydromineral balance. Again, osmoregulation. There are what we call the osmoconformers. This includes the agnathans, the hagfish, family Myxinidae. They're what we call ionic osmoconformers. They actually don't worry about blood water balance because their blood plasma has the same osmolality as seawater, meaning that water is not going to flow down its concentration gradient. It doesn't go anywhere because it's the same concentration gradient on both sides, inside and outside of the hagfish. Another type of osmoconformer are the chondrichthyan fishes. These things produce TMAO and urea, which I'll talk about in the next slide. And what they do is they match their blood plasma osmolality with that of the ocean water. And again, they don't then have to deal with water flowing in or out because they have the same osmolality as the water around them. And then we have the osmoregulators, the teleosts, the bony fishes, osteichthyes. They are fighting this battle where they say, we're going to maintain a body osmolality that's different from that of our environment, and we are just going to constantly pump ions across our gill to fight this. We're going to constantly do it. This includes the saltwater teleosts, again, the hypoosmotic environment, or the hypoosmotic state, and then the freshwater teleosts in the hyperosmotic state. Also the lampreys, because remember, most lampreys are freshwater. So when we talk about osmoconformers. The hagfish is here at the top. An ionic osmoconformer. Primarily, they maintain a very high level of sodium ions in their blood, such that their ions are so high, they have the same osmolality as that of seawater, about 1,000 milliosmoles. Their skin is permeable. They don't really care if water comes in or out because their osmolality is the same as that of seawater, so water really isn't moving in or out of these things. Any water that does diffuse in, the kidney can clear excess water if it does happen. They also can excrete ions in the slime and also in the urine if they need to. For instance, if they're trying to balance particular ions in their body for other chemical reactions. But there's no real active transport of any kind of pumps of ions in the gills, gutter, or skin. They really kind of just secrete it in the slime. So these things basically, their body fluid is seawater. That's how they get away with it. Now, the elasmobranchs produce what's called urea TMAO. And so this figure here shows hagfish. Hagfish, this is the osmolality of salt water, osmolality of fresh water. Hagfish, their osmolality, usually ions, they just match that of salt water, and so they don't deal with osmoregulation. The elasmobranchs are here. So a saltwater elasmobranch is going to have a plasma osmolality. I mean, basically any fish it's going to be about 300. We always say the blood plasma of a living vertebrate is about 1/3 the strength of seawater. And so they are going to osmoconform by adding excess urea and TMAO indicated by this orange box here. And meaning that their plasma osmolality at the end of the day is equivalent to that of seawater, which means osmosis is not going to occur. So they don't have to worry about water moving into or out of them. Now, this TMAO is trimethylamine oxide. It's produced in the liver. And the elasmobranchs then produce urea as a breakdown product of nitrogen metabolism, so breakdown of proteins. Now, if a bull shark, for instance, which we know can live in fresh water, if a bull shark decides to swim into fresh water, what does it do? It just urinates out the excess urea and brings its blood plasma osmolality closer to that of fresh water. That's how chondrichthyan fishes osmoregulate. They also have some ion pumps in their gills, the rectal gland, and the kidneys that then also can help to regulate ion excretion if something here were to fall out of balance. But generally speaking, they just match their blood plasma to that of their environment using urea and TMAO. And they're what we call the osmoconformers. Also, the chimeras, the holocephali, the ratfish also do this. Now, saltwater teleosts are what we call osmoregulators. They are hypoosmotic osmoregulators. Their blood plasma, again, is about 380 to 450 milliosmoles, which again is less than that of seawater, meaning that they lose about 30 to 60% of their body weight per day in water. And this is lost over the gills. That's how fast they're dehydrated. When I say saltwater environment is a desert, that's what we're talking about. They gain salt also by diffusion. Salt's coming in at the gills. So what they do is they drink. What? They drink? Yes, they actually drink. You ever heard the phrase drink like a fish? Well, you're drinking like a saltwater fish. They drink about 7 to 35% of their body weight a day in water. Now, I don't know how many gallons of water that is for a human, but that's a lot. And they absorb 60 to 70% of that water from their gut. And so this is, they actually actively drink seawater. They're drinking all the time. So the gills, the intestine, and the kidneys then excrete excess ions. So they're drinking this water, drinking the seawater, absorbing the H2O out of it, and then excreting the excess ions through their gills or kidney or intestine. So they produce a very small amount of isoosmotic urine because they're retaining water. They don't produce a whole lot of urine because, again, they're dehydrated. Now, freshwater fish are hyperosmotic osmoregulators. their blood plasma is a little bit less dilute than a saltwater fish. Again, 260 to 300 milliosmoles. I always like to say 1/3 of saltwater. But saltwater, is not distilled water. There is solute in fresh water. Sorry, there is solute in fresh water. Usually somewhere between 0.1 to 10 milliosmoles worth of salt. So it's not distilled water. Freshwater fishes must absorb salts and remove excess water because they're constantly flooding. So they gain, talk about water weight gain, guys. They gain 1/3 of their body weight every day. They have to get rid of this. And so again, they came up with the kidney as the master, the water pump. The kidney pumps this water out. They waterproof all surfaces of their body except the gills, which they can't because you still have to have gas exchange there. Otherwise, you'd suffocate. Freshwater fish do not drink. The only water they might ingest is that with a prey item or food item. They absorb salts in the gills and from the intestine. They're actively pumping salt in. They're actively taking it in from their diet. And they resorb any salt in the kidney. So they produce what's called copious amounts of dilute urine. If you ever pull a freshwater fish out of the water and you're looking at it like you caught it and you're taking it off the hook and you squeeze it and it shoots a stream of urine out and you're like, oh, look, this is fish urine. Don't worry. It's 99.99% water. There's hardly anything else in it. These things literally are just excreting fresh water all the time. So freshwater urine, freshwater fish urine is pretty much water, H2O. So here's a figure then that shows that. And if I was a student studying for this class, I might consider committing this to memory because I'm not for sure, but it might be an exam question. These are the ways that the ions in water move. So here's a yellow perch. Family Percidae, Perciflavescens. It's a freshwater fish. They are, here is the heavy white arrows are active, meaning that there is energy being spent to pump. Diffusion is passive, meaning flowing down a concentration gradient. Can't stop it, can't help it. Salts actively pumped in at the gill. Freshwater fish need salt. Water is leaking in passively, and the saltwater or the freshwater fish is losing salt passively to the environment. Down its concentration gradient, down its concentration gradient. These things produce constant, copious, dilute urine. They're constantly urinating. Okay, saltwater fish, drinking like a fish. Here's a bluefin tuna, drinking like a fish. Taking water in, taking the H2O out in the gut, into the blood, taking the salt, pumping it out over the gill, getting rid of that excess salt. But despite our best efforts, we are always dehydrating water passively. We always have salts trying to creep in. So they actively pump salt out, actively drink water. Okay, the textbook has more of an explanation of this, but that is a figure you might want to know. Now, when I talked about the kidney being an invention in freshwater fishes to pump water out, when we think about a major difference between fishes and terrestrial vertebrates such as ourselves, your kidney is designed to pull urea out of the blood and excrete it in the urine. Fishes don't do this, all right? Number one, most fishes produce ammonia as a waste product, not urea. Chondrichthyan fishes make a lot of urea, less ammonia. But most fishes produce mostly ammonia as a waste product and not urea. We produce urea, us terrestrial animals, mammals. Because the kidney is primarily a water pump in fishes, it's not really even pumping out ammonia or urea. The gill does this. The kidney doesn't. The kidney, like here, the dogfish, 0% of the ammonia is excreted by the kidney, right? 7% of the urea is excreted by the kidney. 91% of the urea is excreted at the gill. It goes out the gills. So if anything, if you're holding it, instead of worrying about a fish urinating on you, when you go to hold it up by the operculum to get a photo, the gills are where all the urine's at. That's where all the urea's at. So it's not coming out in the urine. And so if you look down here, in the case, the only fishes really that excrete a lot of ammonia in the urine really are a lot of odd fishes, in this case, wrasses and blennies and coral reef fishes. So like kind of warm tropical marine fishes will do this, warm tropical marine fishes. All the other fishes, their primary metabolic waste product is ammonia and is excreted across the gill. The chondrichthyan fishes, of course, then use urea because they use it to help osmoregulate. So how did they do this? This is a legend for the figure on the next page that is well detailed in your textbook. So this is the chloride cells, also called the mitochondrial rich cells of the teleost gill. Here's our figure. So this previous slide here just is an explanation, A, B, C, D of this here. So A, B, C, D, all these processes. So the first one is this. Figure 1, this one right here. Mitochondrial rich cells have very highly infolded basolateral membranes. They provide a lot of surface area. And this creates a tubule system at the gill. So this is the water side of the fish, external environment. This is the inside of the fish side. And this cell sort of sits in between at the gill. So this is on the gill filaments and the lamellae of the gill, or the lamellar folds. 2, the apical surface of some mitochondrial-rich cells of the gill in freshwater fishes take up chloride from the environment in exchange for bicarbonate. What? So here, there's an ion pump. There's an active process here that is shown by the white arrows, and then there's a passive diffusion process shown by the solid arrows. And what happens is this. I told you last time when we were talking about carbon dioxide likes to be in the blood in the form of carbonic acid, and that carbonic acid was the savior of all life. What happens is carbonic acid forms from the production of water and carbon dioxide. This then carbonic acid diffuses into our favorite compound, bicarbonate, and a proton. That bicarbonate diffuses down its concentration gradient out of the fish into the water, and in doing so, it is exchanged for a chloride. These things have the same negative charge, so we're remaining net neutral. This is why chloride diffuses in. If we lose a negative charge, we got to gain a negative charge and maintain neutral balance. And again, I told you, freshwater fish try to get salt in their body. This is how they do it. They get rid of a metabolic waste product to get their salt. Now, sometimes this proton can be pumped into the blood, but typically speaking, this proton actually is pumped out of the gill. And in getting rid of this positive charge, we bring in a positive sodium. Again, one of the goals of a freshwater fish is to pull salt out of the environment. And sodium chloride is in fact salt. So we can actually use our carbonic acid, pumping it out in the form of bicarbonate and a proton. And in doing so, we accumulate then the salt that we so desperately need to survive in our freshwater environment. Pretty cool. Now, when we look at marine fishes, here's a saltwater fish, it's a lot simpler. I mean, all they do is, again, I said these ion pumps, if a freshwater fish is pumping sodium chloride in, what do you think a saltwater fish is doing? Pumping it the opposite direction. And so this is a freshwater chloride cell. Here's a saltwater chloride cell. Same kind of thing. Lots of surface area here, lots of basolateral infolding, but our pumps are in the different direction. What we're doing is we are actively pumping chloride from the blood into these chloride cells in the gill, and then chloride is at such a high concentration here, it diffuses down its concentration gradient into the marine water around the fish. Now, because we got rid of a negative charge here, sodium that's hanging around in the bloodstream is going to have this leaky junction here between the chloride cell and what we call basement cell. This sodium this positive charge is attracted to this negative charge. And so sodium just literally comes on out and follows chloride. So all we're really doing is pumping one side of this equation. The other side passively goes out. Likewise, over here in the freshwater situation, we're only pumping this proton. This passively goes out. So we're only pumping one of the ions and the second one follows. Okay? In this case, there's an exchange. Don't worry about calcium here. Just worry about sodium and chloride. And so, again, if I was a student studying for this test, you might want to know that biochemistry. So, one quick thing before we go on to our talk. And again, this figure is from your textbook. It's well detailed and explained in your chapter. It actually, your chapter is going to give you a lot more detailed overview than I just gave. And really, as far as the exam, focus on the detail I provided you, not every little detail in the chapter. So there's a few specializations here among fishes. There are fishes that we call stenohaline. These fishes tolerate only small changes in solute concentration in their external environment. So generally speaking, most fish are designed to do this or this. Notice that these pumps are all designed to pump in the opposite direction. Most fish, it's a one-way street. Their gill does this or does this. They really can't switch this. That's actually kind of a rare thing. So things like a bluegill, Lepomis macrochirus, the bluefin tuna, Thunus thunus, and the zebrafish, Danio rario. If you take a bluegill and put it in the ocean, it dies. If you take a tuna and put it in Jordan Lake, it dies. These things only tolerate the environments in which they're designed to be in, freshwater or saltwater. Same with a zebrafish. And I put the zebrafish on here because a lot of times the zebrafish is a research model. It's an organism because they sequence the genome, and these people a lot of times do research on zebrafish. I would not do a study on osmoregulation of the zebrafish because it's stenohaline. It cannot tolerate a change in osmoregulatory conditions. It dies. Now, there are fishes, I don't have the salmon on here, but salmon are another one. They are euryhaline. These things have the unique ability to osmoregulate over a wide range of environmental salinities. This includes diadromous and estuarine fishes. So the example here, striped bass, Morona saxatilis, the Mozambique tilapia, Oreochromis mozambicus, and then the bull shark, Carcharinus lucas. Now, these fish have the remarkable ability to be able to be in a freshwater environment doing this. And then if you take them and put them in a marine environment, they're going to have a little bit of a getting used to. But actually, they can switch their gill over to this in a matter of hours. They can restructure their gill and redesign it, reconfigure it to adapt to what is called an osmoregulatory challenge. So a striped bass can do this relatively quickly. Same with a tilapia and a bull shark. Now, we talked about bull sharks. Bull sharks, of course, are chondrichthyes. They don't have any of this going on. Only the bony fishes like the striped bass and the tilapia do. The bull shark, of course, is going to change the level of urea and TMAO in its blood to match the osmolality of the environment in which it's in, whether that's running its osmolality down closer to fresh water, or whether that's running it up closer to salt water, because bull shark is a chondrichthian osmoconformer. It does not play this osmoregulatory game. It just says we're matching our blood plasma to that of the water around us. That way osmosis doesn't occur. So two different mechanisms of being euryhaline. Salt pumps in the gill or urea TMAO in the blood. But rarely, this is a pretty rare phenomenon. These fishes here are unique in that regard. There's not many fishes that are euryhaline. So the last thing I'm going to talk about here from your textbook is the stress response. And this is the last of your homeostasis mechanisms. It deals with things such as avoidance of predators, disease resistance, things like this. So the stress response is controlled in a combination of nervous and endocrine systems that we opened the lecture by talking about. So there are three stages of a stress response. There are what we call the primary, secondary, and tertiary responses. They're detailed here. So a stress is perceived by the nervous system, and then a response is then made by the nervous system and the endocrine system. And so when we talk about perceiving a stress, that is an afferent signal to the central nervous system. For instance, our eyes see a predator. For instance, presence of a predator is a perceived stress. Or the fish hears a sound that indicates it might be captured. All right? Afferent signal to the central nervous system, the fish understands something is wrong. Or it could be a chemical stressor. For instance, water pollution, low dissolved oxygen. It's swimming along and everything is fine. And all of a sudden it notices, well, wait a minute, it's a little bit harder to breathe here. Something is wrong. High ammonia, water pollution. Or it's been captured, it's struggling, or it doesn't have enough food to eat. So there's several types of stressors. These are all processed by the fish in the central nervous system. Once that process has been made, the fish then sends out an efferent signal out, and then also activates the endocrine system. This is things like adrenaline, corticosteroids, things like that. So the primary nervous system, or the primary stress response rather, stimulation by the nervous and endocrine systems. Perceived stress is received. We understand that. Let's start sending a signal out. Let's tell the body, we got to deal with this. Then the secondary stress occurs. Secondary response includes things like physiological changes. If we are trying to outrun a predator or outswim a predator, we need blood glucose. We need to get glucose to our muscle. We need to get energy to our muscles to swim away. So we might see an increase in blood glucose, for instance. That means that we've got more energy going out. We might see the fish is now swimming and it's actively swimming away. And in order to do this, it has to pass blood across its gills more quickly to get oxygen. It needs more oxygen. But in getting more oxygen, we are exposing our gill to a greater exchange of water and salt. And that throws our blood plasma osmolality off. But we have to get the oxygen because if we don't outswim this predator, we're dead anyhow. We'll deal with osmoregulation later. And then if the stress persists for too long, we have what's called tertiary response. This is changes in animal health. For instance, decreased growth rate, decreased reproductive output. We're dedicating a lot of energy to this stress. For instance, let's say we live in a terrible polluted environment like the Cuyahoga River. We've got to deal with constant water pollution. We're not going to grow as well as a fish in a nice, happy environment. Also, this is important because diseases itself is a stress, and under prolonged stress conditions, our ability to resist diseases also decreases. And so the goal is this. I like to look at stress as the goal is to survive the stress, and all this other stuff can wait until later. So if our goal is to outswim a predator, let's figure that out first, then we can fix this other stuff. So tertiary responses are changes in whole animal health, growth, reproduction, disease resistance, feeding, aggression, whatever. Okay, and it all starts then with the perception of a stressor, which if you go to the stress biology literature, a stressor is defined as a noxious stimulus. Noxious meaning not very nice, undesired. For instance, students, clear off your desks. You have a pop exam is a noxious stimulus. So that's the stress response. And the book also goes over this. But again, just know that this is part of our integrated physiology, and it's part of this regulatory network, including the nervous and endocrine systems. Now, why is this important? I will tell you, and I'll end with the story. Here it is, AEC 441, you learned it here. Stress and fishing mortality. Now, this is sort of tying us all together. Why is osmoregulation important? How is it tied to oxygen consumption? And how is this tied to stress? Well, here you go. When a fish is hooked, let's say you're out fishing, you catch a fish, you've got a fish on the line, it bit, you set the hook, it's on there, struggling. When you hook a fish, it of course realizes something is amiss. It has perceived this noxious stimulus of a hook in its mouth. And it will begin to fight against that line. And you start reeling it in. Now that fish is going to increase its oxygen uptake by increasing the rate at which it ventilates water over its gills. It's going to increase the rate at which it pumps blood into its gills to get oxygen. And then that blood circulates then to the muscles that are working to swim against you, reeling it in. But the drawback then is the more blood you pump over the gills, the more salt and water exchange occurs there. Whether it's a marine fish, you're dehydrating it. Freshwater fish, you're diluting it. But it has to do it because it has to have that oxygen so it can fight you. So the freshwater fish, the blood becomes dilute as the fish takes on water. For saltwater fishes, the blood becomes concentrated as the fish loses water. The fish must rapidly increase the rate then of salt uptake in the freshwater gill or excretion in the saltwater gill to counteract these osmoregulatory changes. But this costs energy. But we're using our energy to try to swim against the hook, to fight. So where do we dedicate the energy? Well, we'll eventually fix this osmoregulatory problem, but it might take some time. So if the osmoregulatory pumping cannot keep pace, the fish will undergo osmotic imbalance. The osmolality of the blood will be thrown off, and the fish will eventually go into shock, and this will lead to death. Now, the longer the fish has to elevate its ventilation rate due to this exertion, the more likely it is to go into ionic imbalance, experience osmotic shock, which is a secondary stress response. Again, here, changes in plasma osmolality. This actually, I'm going to say, is the primary cause of catch and release mortality in fisheries. It is the primary cause of death. It's really osmoregulatory distress, not necessarily suffocation. Although if fish are really crowded against each other, like in the bag of a net, they can suffocate or asphyxiate. But generally speaking, primary cause of death is they struggle so hard to get that oxygen that they put themselves into osmoregulatory distress, and they literally dehydrate themselves or flood themselves to death, depending on whether they're in a marine or freshwater environment. So if you intend to catch and release a fish after hooking it, the best thing to do is just get the fish on the line, reel it in, unhook it, and release it. Don't sit there and play the line, because if you're going to do that, might as well take it off the hook and throw it in your cooler, because even if you release it and it swims away, that fish is probably going to die an hour or so later. Because it might look like it's okay, but if it cannot overcome this osmoregulatory stress, it most likely will die an hour or so later. Or in the least, it's very much vulnerable to predation by something else, because it takes a long time for a fish to recover from this. Even if you're fighting it for 5 or 10 minutes, it might take it several hours to fully recover. Okay, so that is your story. You learned it here at AEC 441. This is stress, fishing mortality, integrated physiological systems, primarily looking at homeostasis and osmoregulation. So with that, I'm going to stop recording, and I will accept and entertain any questions.